Otherwise, we will start, sir.
Okay, we'll wait for another 2 minutes.
Okay, sir. Abdullah?
Yes, ma'am.
Okay. Kavita, are you ready?
Yeah, ma'am.
Okay.
Good afternoon, ma'am. I am daughter of S. Viswanathan. I have also registered for this meeting and this is my second seminar, ma'am.
Okay, very good. Most welcome. Most welcome. Vidullah, I think.
Yes, ma'am. Abdullah.
Good afternoon, all of you.
Good afternoon.
Hello.
Good
afternoon, sir.
This is Krishna Kishore from Narayana Educational Institutions, Hyderabad. Thank you one and all for the organizers for having such a type of webinar, and the topic is also very apt for the present situation. Thank you once again.
Okay, thank you.
Shall we start, ma'am?
Principal, sir, shall we start, sir?
Yeah. Please start.
Okay. Okay, sir. Okay. Kavita?
Yeah, ma'am. A very good afternoon to one and all present over here. I am Dr. Kavita, Assistant Professor of Microbiology, Sadakathullah Appa College. On behalf of the management and the Department of Microbiology, Sadakathullah Appa College, I would like to extend my warm welcome to this international webinar on transmission, pathogenesis of SARS-CoV-2 and its host immune response. So COVID-19 pandemic is not only a threat to our health, but also become a great disruption to our way of life. Nothing in our life is to be feared. It is only to be understood. Now it's time to understand more so that we may fear less. As microbiologists, we hope that this international webinar is actually the need of the hour to discuss these scientific facts in a more precise way. In these few words, let's begin the webinar officially. The first and the foremost thing to begin with is thanksgiving to the Lord Almighty. Let me call Mr. Muhammad Abdullah of 2nd BSc Microbiology to recite few lines from the Holy Quran.
Good afternoon and assalamu alaikum wa rahmatullahi wa barakatuhu. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين صدق الله العظيم. Thank you.
Thank you, Mr. Abdullah, for your recitation. Now, Dr. Janet Rani, Head of the Department of Microbiology, Sadakathullah Appa College, will welcome the gathering. Ma'am, please.
Thank you, Kavita.
Yeah, ma'am.
Recording in progress.
The capacity to learn is a gift. The ability to learn is a skill. The willingness to learn is a choice. With these Brian Herbert quotes, I am starting the welcome address. Respected Principal, Vice Principal, Correspondent and Secretary, the committee members, Dean of Sciences, Dean of Arts, the IQAC Coordinator, Controller of Examinations, Deputy Controller of Examination, Nurse Coordinator, Chief guest of today's program, staff members of our college, other colleges, national, international participants, students, friends, a very good afternoon to everyone. God's abundant blessings bestowed to our college. Our college is celebrating 50th year of Silver Jubilee. Today, 11th June 2021, is the order of the Microbiology Department webinar. The PG and Research Department of Microbiology happily organizing the international webinar on transmission pathogenesis of SARS-CoV, its human immune response. By the guidance of our eminent respected principal and convener of this international webinar, Dr. Mohammed Sadiq sir, his plan of this series of webinar and his suggestions are inevitable to organize this international webinar. On behalf of the Department of Microbiology, I welcome you, sir. The co-convener, the head of the Department of Commerce, the vice principal of our college, Dr. SMA Syed Mohammed Kaja, sir. His encouragement is helpful to organize this webinar. On behalf of Microbiology Department, I welcome you, sir. SARS is a severe acute respiratory syndrome of coronavirus. The pandemic situation is extended for more than 18 months. It is the right time to know more about the host immune response too. In this direction, it's my privilege to welcome the guest of honor and today's resource person, Dr. Nazirullah Rahmatullah, working as an assistant professor in microbiology, Department of Biomedical Sciences, College of Medicine, Gulf Medical University, Ajman, United Arab Emirates. Our chief guest is a very good researcher, very good teacher for having 19 years of teaching experience. He is going to deliver a special talk about the transmission pathogenesis of SARS-CoV and its host immune response. On behalf of the principal, vice principal, correspondent and secretary, the managing committee members, staff and students of our department, I cordially welcome you, sir. I am very happy to welcome the staff members, administrative heads of various other colleges, research scholars, postgraduate students, undergraduate students, and friends. I welcome you all. We had received a very good response throughout the world for this international webinar, nearly 600 registration, including national, international participants, institutions from Tamil Nadu, University of Hyderabad, University of Bangalore, Adam Mishwish University, Poland, Kuwait University. People also have registered and joined now and participating in the international webinar. Definitely, I ensure this webinar will be useful and next few hours will be enjoyable and fruitful for every one of you. Once again, I welcome you all and thank you.
Thank you so much, ma'am, for your welcome note. Then it's my honor to request the principal of our college and the convener of this webinar, Dr. Mohammed Sadiq, to deliver the presidential address. Please, sir.
Thank you, Kavita. Am I audible?
Yes, sir.
Thank you. Thank you. In the name of Allah, the most gracious, the most merciful. If you could count up the favors of God, never would you be able to number them. For God is all forgiving and most merciful, says the Holy Quran. Respected chief guest of this fine occasion, Dr. Nazirullah Rahmatullah, Assistant Professor of Microbiology, Department of Biomedical Sciences, College of Medicine, Gulf Medical University, Ajman, UAE. While I was looking into his resume, a lot of things I learned from him. He is a life member of European Society of Clinical Microbiology and Infectious Diseases, and he is a member of American Society of Microbiology. So a lot of things are actually represented in his resume. Still, I am very proud of him that a gentleman from our area is shining such a very good effort, taking very good efforts to serve the society. It's very amazing. I pray Allah Almighty that you still get more and more laurels in the days to come. Respected Dr. Janet Rani Madam, the head of the Department of Microbiology, respected faculty members of the Microbiology Department, respected Vice Principal of our college, Dr. Syed Mohammad Khaja, respected Controller of Examinations of our college, Dr. Abdul Qadir, respected Deputy Controller of Examinations, Dr. Karnam Taya, respected Dean of Arts, Dr. Hanif, respected Dean of Sciences, Dr. Roshan, and IQAC Coordinator, Dr. Syed Mohammed, and various heads of the department of our college, research scholars, and faculty members from friendly institutions, and participants from other institutions, and my dear students. Really, I feel very happy that the Department of Microbiology is organizing this international webinar on the topic of transmission pathogenesis of SARS-CoV-2 and its post-immune response. It's a wonderful topic, very apt to the current situation, this pandemic situation. As a microbiology student or scholar, we should know these facts. I express my heartfelt thanks to the organizers, the Department of Microbiology faculty members, for their relentless job for the last 1 or 2 months to make this event a great success. And I once again pray Allah the Almighty for having this beautiful chance of grouping together, even in this pandemic situation, to share knowledge, to improve our knowledge in the path of serving the society. Thank you very much.
Thank you, sir.
Thank you so much, sir, for your valuable remarks. So now it's my privilege to request our Vice Principal and co-convener of this webinar, Dr. Syed Mohammad Khaja, to facilitate this program. Please, sir.
Good afternoon and Assalamu alaikum. The Almighty will show the peace and mercy to all of us. I begin in the name of Almighty, the most gracious and most benevolent. Respected Principal, Dr. Muhammad Sadiq. Respected resource person, is a globally known person, Dr. Nazirullah Rahmatullah, the Assistant Professor of Microbiology and Immunology, Department of Biomedical Sciences, College of Medicine, Gulf Medical University, Ajman, UAE. Respected PG and Research Department of Head, Dr. Janet Rani, respected IQAC Coordinator, Dr. Syed Mohammed, respected Dean of Arts, Dean of Science, respected faculty members, Mr. Dr. Senthil, Dr. Sitra, Dr. Feroz Khan, Dr. Manogar, Dr. Hamidullah Sarif, Dr. Surya Prabha, Dr. Kavida, and respected Controller of Examination and Deputy Controller of Examination, and also the faculty members, scholars from other colleges and other countries and other states, participants and students. I am very much happy to participate at this international webinar on transmission, pathogenesis of SARS-CoV-2 and its post-immune response, being organized by the Department of PG and Research of Microbiology, and to offer my presentation on this occasion. I congratulate the department for having selected this suitable topic for discussion. The topic of webinar is more relevant and highly useful to everybody at this unprecedented time due to an outbreak of the second wave of COVID-19 and ongoing outbreak of the pneumonia caused by a novel coronavirus currently designated as the severe acute respiratory syndrome coronavirus 2. SARS-CoV-2 was reported recently. The COVs are the coronavirus are the members of the family coronavirus. The enveloped viruses possesses extraordinarily large single stranded RNA genomes ranging from 26 to 32 kilobases in length. It can cause respiratory, digestive, and nervous system diseases in humans and many other animals also. I take this opportunity to thank our respected principal, head of the department, and faculty members of my PG and Research Department of Microbiology, and the distinguished resource person for having provided this platform to impart the knowledge of SARS-CoV-2 and also its effects. I appreciate the faculty and the organizer who associate themselves with the dedication and supporting the ventures of the webinar. I felicitate our beloved participants, students, and wish every one of you good luck and success. And thank you, Punanda. Thank you.
Thank you, sir, for your kind words. Now, next is the introduction about our chief guest. Let me request Dr. Chitra, Assistant Professor of Microbiology, to give a short
Dr.
brief
Kavita.
note
Good
about
evening.
the credentials of our chief guest. Please, ma'am.
Thank you, ma'am. Good evening, all. I extend a warm welcome to all to the international webinar on transmission pathogenesis of SARS-CoV-2 and its host immune response, organized by PG and Research Department of Microbiology, Sadakathullah Appa College, Tirunelveli. I feel immense pleasure in introducing our resource person, Dr. Nazirullah Rahmatullah. We are thankful to you, sir, for taking the time out of your busy schedule to attend the webinar. He has completed Master of Science in Microbiology from Bharatdasan University. He pursued his PhD in Microbiology from Bharatdasan University. He has 19 years of teaching experience. At present, he is working as Assistant Professor, Department of Biomedical Science, College of Medicine, Gulf Medical University, Ajman, United Arab Emirates. Formerly, he worked as Head and Assistant Professor in Department of Microbiology and Immunology, Misrata University, Libya. Currently, he is doing research on immunogenicity and efficacy of Sinopharm vaccine administered to UAE residents sanctioned by GMU UAE. He has published so many research articles in national and international journals. He is appointed as foreign examiner for PhD thesis evaluation in Dr. NGP Arts and Science College, Coimbatore, and Meenakshi Academy of Higher Education and Research, Chennai. It is an honor and a privilege for us to have you, sir, with us today. I'm sure his presentation will enrich our knowledge on SARS-CoV-2. Once again, I welcome you all. Thank you.
Thank you.
Thank you, ma'am, for your precise account about the hero of our seminar. Now, with no further delay, let me hand over the session to our honorable speaker of this afternoon, Dr. Nazirullah Rahmatullah, Assistant Professor of Immunology and Microbiology, Gulf Medical University, UAE. Please, sir.
Hello. Good afternoon to everyone. Am I audible?
Yes, sir.
Yes,
Can you
sir.
hear me? Yeah.
Yes, sir, you are audible.
Thank you very much. So thank you very much to honor me to invite me to give one-day seminar on transmission pathogenesis of SARS-CoV-2 and its post-immune response. I don't want to waste my time to discuss most of the things. Let's go to the seminar. Yeah. So today the topic is a transmission. It's a catastrophic. The world is completely upside down because of the one small tiny particle, the viral particle, SARS-CoV-2. Isn't it? So you know very well about that, isn't it? So SARS-CoV-2. Yeah. Sorry. Yeah. SARS-CoV-2 is caused by COVID-19. The SARS-CoV virus causes the disease COVID-19. So the SARS-CoV-2 has infected millions of people throughout the world and killed hundreds of thousands people. Nearly up to date, 3.8 million people has died throughout the world in 2020. Is an infection causes the COVID-19, which ranges in presentation from asymptomatic to fatal. So the vast majority of the infected individuals experience mild symptoms like a fever, cough, malaise, myalgia, and headache, taste and smell disturbance, and that do not require hospitalization. So the people recover. They recover very fast within 2 to 3 weeks. A significant number of patients develop severe illness, which is postulated to be related to both either overactive immune response or virally induced pathology. So what we know about these things is, you should know, excuse me. Yeah. So yeah. So SARS-CoV-2 is an enveloped beta coronavirus. It's a genetic sequence very similar to SARS-CoV-1. So the coronaviridae, you can see this coronavirus comes under the family coronaviridae. So the coronaviridae has four subgroups: alpha coronavirus, beta coronavirus, gamma coronavirus, delta coronavirus. This SARS-CoV-2 comes under the beta coronavirus. In that group, SARS-CoV-1 and 2 is there. So the SARS coronavirus 2 is very similar, close similar, genomic sequence very close similar to SARS coronavirus 1. It's nearly 80% they are sharing the genetic sequences, nearly 80% they are sharing to COVID-1. And nearly more than 96% genomic sequences are very close with the bat coronavirus that is called rat G13, isn't it? So this virus first isolated in the bat in China. That's why they have given the name raw TG13. So the coronavirus, you know about, all of you know about the coronavirus is made up of enveloped or a single stranded RNA virus. It's an enveloped virus. The envelope contain spikes protein and as well as envelope protein, membrane protein, all are there, isn't it? So the first step in infection is the virus, how the virus bind our body. It's binding to our body cell through the target receptor. The main receptor is ACE2 receptor, that is angiotensin converting enzyme to receptor. So the viral surface protein, this is the viral surface protein, spikes protein, isn't it? It will go, it will bind with the angiotensin converting enzyme 2. See here, you can see here. So this is the gene similarity, and then this is the group. 4 groups are there: group 1, group 2, group 3, alpha, beta, gamma. Isn't it? SARS coronavirus comes under the beta group, and more close related to the beta coronavirus. Isn't it? Here, afterwards, lot of variants they have identified. Till now, nearly more than 150 SARS-CoV variants they have identified throughout the world. Isn't it? So alpha B117 from UK, and beta B1351 South Africa, Brazil, Indian variant, and US variant, and another one B1526, that's this variant, and Kappa B16171 Indian variant, B16173 Indian variant, and Denmark variant, Mint variant. Many variants they have identified. Lot of variants are coming. We don't know how this variant are forming, maybe due to the climatic conditions or something like that. Still it's a puzzle, isn't it? So why we have to know about the SARS coronavirus, isn't it? So the SARS coronavirus 2 is more infectious than the SARS coronavirus 1. Why? So the SARS coronavirus 2 has a high reproductive number when compared with the SARS-CoV-1. Isn't it? So indicating much more efficient spread to the people to people, while both SARS coronavirus 1 and as well as SARS coronavirus 2 preferentially interact with the angiotensin converting enzyme 2 receptor, isn't it? So it has a structural difference in its surface protein that enables stronger binding to the ACE2 receptor when compared with the SARS-CoV-1, COVID-2. It has a stronger binding attachment on this ACE2 receptor because of the surface protein contained RBD receptor binding domain is there. That domain, it has more attractive and then bond affinity to the ACE2 receptor and greater efficacy at the invading the host cell. So especially it is more affinity on the AC receptor, which are present on the upper respiratory tract and the conjunctiva. So the SARS-CoV-2 load is higher and peak is observed at the time of symptoms onset in the first week of illness, with a subsequent decline after that. So it indicates that the highest infectious condition, highest infectiousness potential just before or within the first 5 days of symptoms onset. Those who are having symptoms, the first 5 days, the starting of the symptom, during the starting of the symptom or within the 5 days of the infection, and that person, he will be more highly infectious. He can spread the virus to many people like that. So that's why the WHO and then government and then government organization, they are insisting the people we have to maintain the distance, social distance. So, but in contrast with the SARS-CoV-1, the highest viral loads are detected in the upper respiratory tract at the second week of illness. So that's why because when compared with the SARS-CoV-2, so COVID-1 is lesser bond binding with the ACE2 receptor. So when compared with the SARS-CoV-1, SARS-CoV-2 is highly contagious, isn't it, in the first week after the symptom onset. Yes. Then you can see the mode of transmission, how the viral particle transfer, isn't it? So the mode of transmission usually through the contact, through the droplet nuclei, airborne, vomits. Now recent studies, they said the particle we may transfer through oral fecal and as well as urine droplets, as well as some studies they have said, but there is no evidence at all, the bloodborne and as well as animal to human. Animal to human is there, but other SARS viruses like a rat G13 viral particle is there. But there are the many members, they can spread the SARS virus. For example, MERS virus, Middle East syndrome, respiratory syndrome virus. Many viruses are there, SARS group viruses. They can transfer from animal to human. The primary mechanism of transmission is via droplet nuclei. You can see here. So infected people, SARS people, so through the cough droplet. If the cough droplet is lesser than 1 micron, it may travel more than 1 meter, 5 micron, lesser than 5 micron. If the respiratory droplet is more than 5 micron, more than 5 micron, it may transfer less than 1 meter, like that. So the droplet cough droplet nuclei transfer through inanimate objects, even in the restroom, through the airborne, or through the inanimate object, cell phone, or countertop, or glass cup, or door handle. It may be transferred through many inanimate objects. The target of the host receptor, I'm not able to see it. The target host receptors are found mainly in the human respiratory tract epithelium, including oropharynx and the upper airway. The conjunctiva and the gastrointestinal tract also be susceptible to infection and may serve as transmission portals. The transmission risk mainly depends on such as the contact pattern, how we contact with the people, very close contact or very gathering, like that. So based on the contact pattern, the transmission risk will depend, and then based on the environment and the infectiousness of the host and the socioeconomic factors play a major role, because in so many poor countries they could not able to afford the mask, and then could not able to afford the hand sanitizer. They could not able to maintain the social distance because of the poverty, and the socioeconomic factor play a major role to spread the disease. For example, in our place, so how long we can maintain the the quarantine, how long we can maintain the lockdown. So afterwards, we have to be released the lockdown, because otherwise the poor people will suffer. So for that, the socio-economic basis are play a major role to transmission risk of the SARS-CoV-2. The most transmissions occur through the close range contact, such as 15 minutes face-to-face or within 2 meter distance. So the people, they may acquire the infection quickly. The spread is especially efficient within the household area through gathering of the family gathering and friends. So also the aerosol transmission can still factor during the prolonged stay in crowded areas or poorly ventilated indoor settings, especially the marriage hall or in the vegetable market areas or in the bus travel, train travel, even flight travel. So that is the aerosol transmission still can still the factors, one of the factors to transfer from one people to other people. Then SARS-CoV-2 genome, it comprises nearly 14 open over a frame is there. So out of the 14 frame, 2/3 of the frame which encodes 16 non-structural proteins. It's a not important non-structural protein that make up for the replicate complex and other things. So other replicative activities, releasing activities, non-structural proteins. These are the proteins are synthesized from the 2/3 of the open reading from nearly 16 non-structural proteins are there. But 1/3 of the ORF frame encodes nearly 9 accessory proteins are there. So, and 4 structural proteins are there. That's 4 structural proteins are very important. One is a spikes protein, envelope protein, membrane protein, nucleocapsid protein. So, of which the spikes protein mediate the entry of the SARS-CoV-2 in our body cell. So, this is the ORF region structural protein, especially spikes protein. Yes, sir. So how the viral particle enter in our body, isn't it? So this is the ACE2 receptor. ACE2 receptor normally present in the oropharyngeal region, lung region, gastric intestinal region, as well as gallbladder region, as well as in the testis region. Many areas it is there. So ACE2 receptor is there. So if the viral particle enter in our body, it can attach on the ACE receptor because the ACE receptor act as a receptor to bind with S protein, because the S protein contain RBD protein. That RBD protein is compatible with that ACE2 receptor. Once it attached, it go for endocytosis. So which is it go for the endocytosis and form the endosome. So once it form the endosome inside the cathechin L lysosome is play a major role to tag the viral particle and release the viral genome. That is single stranded positive sense RNA is there. This positive sense RNA immediately bind with the ribosome and go for the non-structural protein synthesis and then go for further replication. By using further replication, they can synthesize their own genome, that is called the baby genome, progenome, and then afterwards they can once again synthesize the secondary structural protein for proteins. Those are nucleocapsid protein, spikes protein, membrane protein, envelope protein. All the proteins will be attached in the endoplasmic reticulum. Then the endoplasmic reticulum combine with the Golgi complex and form the endoplasmic reticulum Golgi integrated complex, and then the baby viral particles, that means progeny viral particle, will form and then release from the cell. While releasing, most of the cells will be lysed because of the osmotic imbalance and then arrest the metabolic activity due to the metabolic accumulation, due to the toxic molecule accumulation, cell will be destroyed. Yes. So what are the main the clinical presentation of the SARS-CoV-2 pathogenesis? Though the viral particle enter in our body and multiply, but what are the main clinical presentation of COVID-19? Usually, most of the people, they have the sinusitis, like a sinusitis, rhinitis, anosmia, pharyngalgia, rhinorrhea. This is the common, and along with that sore throat. This is the common symptoms like in flu fever or common cold symptoms. Isn't? But one symptoms are two are very peculiar. That is called ageusia. That is a losses of taste, and then along with that is a smell, losses of smell. Isn't? This is the typical symptoms of the SARS-CoV-2. And then lung complication will be there. Usually the cough will come, dyspnea is there, expectoration, chest pain is there, then leads to pneumonia, and then ARDS is an acute respiratory distress syndrome will form. So if the viral particle multiply, then further complication will be there, especially in the heart, arrhythmias, chest pain is possible, even neurological symptoms, dizziness, headache, impaired consciousness will also be possible in many cases. And then most of the cases in the asymptomatic to mild cases, most of the people, they may have myalgia, arthralgia, malaise is there, and then back pains are usually common. Some people, they have digestive complications, digestive problems like a diarrhea, nausea, vomiting, abdominal pain. These are the things that are normal in the COVID clinical manifestations. So mainly systemic symptoms, usually body chillness, fatigue, malaise, and fever. This is a quite common. Some people, they may have fever for 1 or 2 days. Some people, they may have more than 10 days like that. It depends on the body condition and on the viral multiplication and immunological process. Here, I want to talk about the immune response. This is the main one. So how our body responds to the COVID-19? It's mainly based on the body nature. So you should understand what is the immune response to the general pathogens. In our body, there are two immune responses are there. The role of immune responses, two major immune responses are there. One is a humoral mediated, that is antibody mediated immune response. Another one is cell mediated immune response. In these two immune responses, that is the major immune responses, these two are the adaptive immune responses. Innate are there. Innate also will play an important role to eliminate the pathogens, not only the viral particle, not only the COVID-2, even influenza, common cold, or orthomyxoviruses or paramyxoviruses, respiratory syncytial viruses, any kind of viruses, even in the dust particle enter in our body, isn't. So innate immune response will play a major role to eliminate the viral particle, eliminate the pathogens, eliminate the foreign materials, isn't. So the immune response to COVID-19, so there are two important things are there. One is humoral mediated and cell mediated immune response, isn't. So this immune response is against to the pathogenesis of SARS-CoV-2 viral particle. This immune response will give the longer term protective immunity or short term protective immunity. It's currently poorly defined. So no article explained, no article clearly stated that the immune response is maximum up to 8 months. Even the people got the vaccinated, even the vaccination, after the taking vaccination, so we can expect maximum 8 months, isn't like that, because there is no available data about the long-term protective immunity against the COVID-19 infection. So here the humoral immunity it's mainly based on the antibody-mediated immune response. If the viral particle recognized by the B lymphocyte will synthesize, will go for activation and synthesize the antibody. That antibody will interact with the viral particle. Then another one is cell-mediated immunity. The cell-mediated immunity is the mainly cytotoxic T cells and phagocytic cells. They play a major role to recognize the viral particle, and then these cell T lymphocytes will activate and synthesize cytotoxic T cells. That cytotoxic T cell will come and recognize the virally infected cell, then try to collapse, try to destroy the virally infected the whole cell. So now, what are the major cells involved in the immunological response? Especially lymphocytes play a major role both in the humoral immune response as well as cell-mediated immune response. What about the memory capacity? Whether the immune cells has memory? Yes, of course. Our immune system has memory cell. Like how we are taking selfie to memorize our photos, our close friends photos, our family photos. After long 10 years back or after 20 years back, we will remember those pictures and other things. Same like our body has memory cell. Once the viral particle, not only viral particle, any pathogens enter in our body, so the memory cell will memorize that antigen structure. If the chance is there, if the same viral particle or any pathogens, any foreign material or any antigen enter in the next time or second time, immediately the memory cell will memorize that antigen structure, then immediately start to synthesize antibodies and respond to the immunological response to that particular antigen. So that is the activity of memory cell. So the lymphocytes it's here the critical important to understand if SARS-CoV-2 infected individual who recover from mild disease develop functional immune memory cells and capable of protection from subsequent SARS-CoV-2 infection. So what are the memory cells are present in our body? Mostly monoclonal B memory cells is the monoclonal B memory cell, and then CD4 T cells, CD8 T cells. These are the cells that play an important role in the memory capacity. Out of the memory B cells play a major role to synthesize a large amount of antibodies and other things. So if the people infected by SARS-CoV-2, whether they have memory capacity, sure they have the memory capacity, but the big question is, $1 million question is whether that memory cells are short-lived memory cells or long-lived memory cells. This is the research is going on. So our project is based on this one. After taking vaccination, so how long the memory cells are persist in the vaccinated people. So here the question is there, isn't it? So after the COVID-2 infection, the infected people are fully recovered from the COVID-2. The functional immune memory cells, how long persist in their body. So first of all, you should know the immunological memory is primarily mediated by the cells of adaptive immune system, and not in the innate immune system. So during the adaptive immune system, the B lymphocytes and T lymphocytes play a major role to control the infection, to eliminate the infecting particle. So they are using many weapons. They are using B lymphocytes and T lymphocytes. They are using many effector molecules. Those are antibodies, interleukins, tumor necrosis factors, and interferons to control the infection. Here the problem started. So what is the problem? In the ordinary people, asymptomatic people or mild people, they have limited or is sufficient antibodies, interleukins, and along with the effector molecules play a major role to control the viral particle. In some people, they are in the normal mild, after they are going to recover, but suddenly they got severe immunological responses. That is called cytokine storm, isn't it? We will discuss in the next slide. Okay. So here you should know what is this one. So how the viral particles are recognized by our immune cells. For example, if the viral particle attached on the AC2 receptor of respiratory cells, isn't it? So what in the oropharyngeal region or upper respiratory tract region, once it attached on the AC2 receptor, then the viral particle will enter inside the cell through the endosome. Then it will be peeled by cathepsin lysosome. Then naked RNA will be released. Then the RNA will go for multiplication, and then further multiplication, the progeny viruses will be released. So during releasing of progeny viruses, some of the viral protein, especially spikes protein, will be expressed on the cell surface. During that time, alveolar macrophages, that is mainly present in our lung, alveolar macrophages come and recognize what are the antigens are present outside the cell. They can recognize and then take up the protein and then internalize that protein, S protein, and then the alveolar macrophages will express those protein with the help of MHC2. So here the MHC2 is a player major role to express the antigen. Without MHC, no cells will come and recognize the expressed protein, isn't it? So alveolar macrophages act as an antigen presenting cell to present the viral protein which is coated on the infected cell. So with the help of MHC2, the viral proteins are expressed. So then the alveolar macrophages, they immediately secrete the molecule is called interleukin, cytokine interleukin 1. That interleukin 1 will give the signal to T helper cell. Now the T helper cell will come and recognize the viral antigen with the help of PCR, T cell receptor. But how they come and recognize that viral antigen, the major receptor, major CD molecule is called the CD4 molecule, which is present on the surface of T helper cell. And that CD4 will come and recognize, check the MHC2 molecule. If it is compatible, then the T helper cell will come and bind with the viral particle. If the CD4 and MHC2 molecule is not compatible, the T helper cell never recognize the viral particle. Even other signal, maybe more than one interleukin, may also induce the T helper cell. They never recognize the viral antigen. For the viral protein or viral antigen recognition, T cell receptor CD4 should be must, and along with that CD3 molecules. These molecules are play a major important role to recognize the viral antigen. Once the T helper cells successfully recognize the viral antigen, then what will happen? The T helper cell will synthesize interleukin 2. That interleukin 2 will induce the B lymphocyte. These B lymphocytes, which are present on our peripheral blood area. So usually the B lymphocytes are immature. That immature B cell is triggered by helper T cell. This helper T cell, once it trigger the B lymphocyte come and recognize the existing viral antigen are present are freely available viral antigen or the progeny viral particle. So wherever the antigens are there, S protein antigens are there, B lymphocyte come and recognize the antigen. According to the antigenic footprint, the B lymphocyte will express the immunoglobulin, IgM immunoglobulin. That IgM immunoglobulin will take the footprint of antigen. According to that antigenic structure and then size and structure, the IgM will be synthesized from the B lymphocyte. That is called maturation of B lymphocyte. So once the B lymphocyte mature, that mature B lymphocyte will differentiate into plasma cells and memory B cells. Those plasma cells will synthesize the exactly same what IgM recognize the antigen, that IgM will be synthesized by the plasma cells. But here the plasma cell durability, lifespan of the plasma cell is only maximum 3 days or maximum minimum 3 days. The lifespan is nearly 3 days or maximum 5 days, isn't it? So sometime, yeah, it is a 3 days to 5 days is there. Is a memory cell. Here the memory cell. This memory cell come and recognize the viral antigen and then kept quiet. They will be in the rest because they memorize the viral structure or the antigen, the viral antigenic structure. This is the memory cell. Now the plasma cell will synthesize the antibody. That antibodies, this plasma cell is called as a antibody producing, antibody synthesizing industry or factory, because the plasma cells are bigger than the plasma lymphocytes. They have the large Golgi complex to synthesize enormous amount of immunoglobulin. The initial immunoglobulin is IgM, immunoglobulin M molecule. It's a pentameric IgM. It's going to interact with the viral antigens and then capture the viral antigens, like how the magnet attract the small soil particle and small ion particle like that. The antibodies will go and capture the viral particle and try to neutralize the viral particle. Once the antibodies are covered the viral particle, then afterwards the viral particle could not be able to attach on the receptor of other fresh cells. Yes, and this is one type of mechanism is there. Next one is there, the antigen, viral antigen presentation by another one method of presentation. So this is this kind of viral particle recognized by alveolar macrophages, and then antibodies will come and attach with the freely available viral particles. So what about the infected cell? If the viral particles are hidden inside the infected cell, that time the infected cell itself will express the viral antigen with the help of MHC1. During the viral replication, the infected alveolar cell, our lung cell, will express the MHC1. That MHC1 will capture the internal viral proteins. That proteins will be expressed on the outside of the cell with the help of MHC1. So during immunological response, any kind of immunological response against any kind of pathogens or any kind of antigens, even the cancer cell neuralization, cancer cell killing, cancer cell killing. So the cells will express the MHC1 or MHC2 molecule. It's very important for that reason. So during any intracellular infection, not only the viral particle, even in the intracellular parasites or intracellular bacteria, any intracellular infection, the infected cell will try to express the MHC1. With the help of MHC1, the viral particle or infected pathogen proteins will be expressed on the outside. Now, this MHC1 and will be expressed here. You can see the T helper cell will synthesize interleukin-2. That interleukin-2 will induce the B lymphocyte as well as cytotoxic T cells. This cytotoxic T cell will come and recognize the MHC1 protein. So the MHC1 protein is holding the viral protein. So here you can see the antigen recognition, and the same viral antigen, S protein antigen, recognized by another T lymphocyte that is called the CD8 T lymphocyte, with the help of CD8 marker. This CD8 marker is very compatible, close compatible with the MHC1 molecule. Once the cytotoxic T cell, CD8 T cell, recognize the MHC1, and then T cell receptor will recognize antigen, viral antigen. Once it attached, then the cytotoxic T cell will go for activation. So how they will activate that? So here you can see the cytotoxic T cell binds to the infected cell with the help of the antigen and as well as TCR and CD8 molecules. Then once it attached and the infected cell, then cytotoxic T cell will release the perforin molecule. Along with the perforin molecule, is a granulysin will be secreting, will be activating, and then this molecule will enter into the infected cell. So the enzyme granulysin and as well as granisine, they will release, and then the granisine enzyme and then perforin molecule make a hole on the infected cell and perforate the infected cell. So during the perforation, the cell will be ruptured, cell membrane will be ruptured, then interferon will enter inside the cell. This interferon inhibit the viral replication. So what will happen, slowly the granisine enzyme make a perforation hole on the infected cell. Now you can see the cell content will comes out, the cell will be destroyed. And as well as viral infection, viral replication will also be blocked by interferon. So nearly interferon alpha, beta, gammas are there. Gamma interferon is very mainly secreted by cytotoxic T cell. So with the help of interferons gamma, granisine, granulocyte, and perforin, all will leads to apoptose the infected cell. So this is the one. This is the main mechanism. A short brief mechanism of immune cells, because if you explain more immunological responses, how the immune cells destroy the viral particle, destroy the infected cell, and then neutralize the viral particles. Many mechanisms are there. I don't want to discuss more detail about the mechanism. It will take a long time to understand the entire immunological mechanism. So that's why I have given the brief idea about how the viral particle enter and recognized by T helper cell and cytotoxic T cell. So here the cytotoxic T cell play a major role to kill the viral particle, not kill the viral particle, and also entirely kill the destroy the infected cell. So what will happen? Our infected cell will be destroyed by our immune cells. Listen. Here, one of the biggest understanding question of the COVID-19 pandemic situation, the pandemic, is why symptoms vary so much among the patients. It is very the symptoms are vary from people to people, isn't it? So I got the COVID infection in the month of January, but I got very mild symptoms. Here in our university, in the month of November, December, January, so many cases were there. But most of the people they had very mild symptoms. I got asymptomatic, very mild and asymptomatic. Some people very severely affected by the COVID-19. The symptoms are very worse, and then they have recovered. In the God grace, they have recovered. So some patients have no symptoms at all. Excuse me.
Sir, one moment, sir.
Okay, sir. Is it okay now?
Yes, sir.
Yeah, thank you, ma'am. Sorry. So one of the biggest question throughout the world, from US to Japan, so top to bottom, and the north to the south pole. So the entire world, they have the $1 million question is there. One is how the symptoms vary from people to people. So some patients, they don't have any symptoms at all. Some people mild, others are extremely severe, isn't it? So among the most severe cases, a common pattern of the disease progression happen like this. The patient gets through the first week with some sign of recovery. Suddenly, then rapidly deteriorated. How they rapidly deteriorated? So in some cases, they require ventilator within 24 hours. So this pattern often in young people and otherwise healthy patients has baffled the doctors. So what causes these patients to suddenly crash, not collapse, crash, isn't it? So the main research is going on. So research now suggests that the patient's own immune system may be to blame. So to whom we have to blame? We can blame the social distancing, and those who are not maintaining the social distancing, those who are not maintaining the mask, and not like that. We should blame our immune system. So many cases, the dead cases, they had a lot of immunological complications. That immunological complications are mainly induced by their own immune system. So the recent research suggests that the patient's own immune system is the blamable. It is called a cytokine storm or cytokine release syndrome. This is new. Is it new? This cytokine storm is new to the people. So for public, it's new to the people. But this kind of cytokine storm is more common in many diseases. So next slide, I will tell about. I will discuss about the cytokine storm. So when our immune system is under attack, yes, and our immune system under attack to the viral infection, bacterial infection, any kind of infections, cytokines play a major role. Yes, and the cytokine, especially interleukin 1, interleukin 2, interleukin 6, and 10, tumor necrosis factor alpha. These are the molecules they are released to recruit more immune cells to fight against the viral particle, to fight against any kind of pathogens. This is a necessary response to defend our body, our immune system from the viral infection. But in some cases, the surge of cytokines cannot be turned off. Listen. So this is the problem. So after the recovery, the viral particles are completely killed. Then the immune systems will be completely turned off. But in some cases, that immune system cannot be turned off. So causing immune cells to invert and damage healthy cells, healthy tissues. So what will happen? Our healthy tissues will be harmed by our own immune system than the viral itself. So that cytokine storm often trigger ARDS, that is acute respiratory distress syndrome. It's a type of respiratory failure. So what kind of ARDS? It's a fluid accumulation in the lung alveolar areas, isn't it? So air sac will be filled with the fluid. So essentially drowning the patients. So many studies, a lot of studies suggest that approximately 40% of ARDS people, they don't survive. So here, what is cytokine storm? You can see the cytokine storm here, isn't So cytokine storms are not new. It's nothing new, isn't it? They have been known to rise from variety of infections, non-infectious illness also. For example, we come to know this during the COVID situation, SARS-CoV viral infection, we know the cytokine storm. But MERS virus, Middle East Respiratory Syndrome virus, causes a cytokine storm. Even influenza, H1N1 viruses, and bacterial infection, especially intracellular parasite like chlamydia, then legionella, many kind of bacterial infections. Even multiple sclerosis also causes cytokine storm. Pancreatitis and primary hemophagocytic lymphohistiocytosis. These also, it's like a type of malignancy. It's not a malignancy, more proliferation of phagocytic cells and lymphocytes in the tissue areas. So this kind of illness, infectious and non-infectious illness may also lead to cytokine storm. So what is the main indicator of cytokine storms? So how we know these cytokine storms are there? The cytokine storms are elevated cytokines levels and increased level of neutrophil and neutrophils when compared with the lymphocytes. So the neutrophil to lymphocyte ratio, NLR, is more higher in the blood. So here you can see the picture. If the woman or a patient infected by COVID virus, the viral particle will multiply in the lung region, especially in the alveolar region. Then what will happen? The fluid will be accumulated. This is called pulmonary edema. Due to the pulmonary edema, the capillary permeability and the blood capillary permeability will be higher, will be raised. Now the viral particle will attach on the ACE2 receptor. The ACE2 receptor is a molecule, it's an enzyme angiotensin converting enzyme 2. This enzyme 2 play a major role to convert the angiotensin 2 molecule into angiotensin 1 to 7, because this angiotensin is a is a vasoconstrictor to dilate the blood vessels, isn't it? So when the viral particle, not only this one, isn't it, the AC2 receptor molecule will also be not available, isn't it, to convert the angiotensin 2 to angiotensin 1 to 7 molecule, isn't it? So what will happen? So if the angiotensin 2 is not converted, the blood vessel will be constricted, isn't it? Another one is, along with that, you can see the macrophagic cells. So the alveolar macrophagic cells, meanwhile, the alveolar macrophagic cells will express the tumor necrosis factor alpha interleukin 6, and and then relative oxygen species will be higher, in the lung region. So these will also leads to blood vessel damage, and along with that, one of the cell is the T helper cell 17. So during this situation, T helper cell 17 will be higher. That T helper cell 17 will induce the T regulatory cells, T lymphocyte regulatory cells. The T lymphocyte regulatory cells down-regulate the CD8 molecules, CD4 molecules. So in the normal healthy people, or in the around 40-50 age people, during the infection time, a T helper cell, that is a CD4 T cell, CD8 T cell will be dropped down, and then T helper cell 17 will be higher. So these are the immunological changes. What will happen? Massive release of cytokines are there. These cytokines will induce the more blood vessel permeability, or blood vessel constrict. So what will happen? So platelets formation, aggregation, like a thrombosis formation is possibilities are there. Along with that, and this cytokine storm and then complementary molecule play a major role to damage the blood vessels. This is the one cytokine storms are there. How the cytokine storm and lymphocyte changes in the mild to severe SARS-CoV-2 infection. If the viral titer is a low, so this low viral titer particle will be eliminated by our immune cells, like a monocyte, macrophages, alveolar macrophages, So what will happen? The normal immune response is quite enough to clear the viral particle. The people will be recovered soon, but sometimes viral load may also play a major role. Heavy viral load will affect the cell, then the cells will be affected. Then, so along with that, cytokines don't play a major role. Especially, you can see these cytokines, IL-6, IL-10 interferons, colony stimulating factor, RANTES protein. These are all the molecules play a major role to recruit more neutrophils and then down-regulate the CD4, CD8 T lymphocyte activities. So due to this one, higher neutrophil, that will damage the vascular tissues, and then lung tissues also will be damaged. So due to that reason, the people will get hypoxia and immediately admit in the ICU. So sometime, if not possible, could not be able to treat the patient could not be able to recover, so leads to death. This is the cytokine storm play a major role, leads to death of the patient. So recent studies reveal that cytokine storm indicators are present in the subgroup of patients. So cytokine storm indicators are there. What are the cytokine storm indicators are there? For example, IL-6 also one of the indicator, interleukin 1 is one of the indicator, and then tumor necrosis factor alpha is one of the indicator. These are the indicators we can identify from the patient blood. If the level is, if the interferon or cytokines levels are more, we can come to the conclusion in that patient we're going to get cytokine storm. So we have to prevent that cytokine storm. So this suggests that cytokine storm may be the reason some COVID cases are worse than the other. So how to treat the cytokine storm? So in the allopathy, in the normal allopathy, so English medicine, like that, like our entire world medicine, so how they are treating the cytokines? Immunosuppressive drugs. No, there is no alternative one. Immunosuppressive drug, corticosteroids are there. So the immunosuppressive drugs are broad anti-inflammatory drugs that reduce the overall immunity. So we want to reduce, there is no alternative choice. We have to reduce the immunity. So we don't bother about the other things. But problem will come if you go for immunosuppressive drug, isn't it? Maybe the fungal infection will come in the unhygienic environment. If the patients are there in the unhygienic environment, in the polluted area, so possibilities are that they can get a fungal infection that is called the black fungal or white fungal or yellow fungal. Any fungal, if they enter, the fungal will also multiply along with the coronavirus. Or after the recovery, the immunosuppressive levels are there, then the fungal infection possibilities are there, isn't it? So not only fungal infection, even the opportunistic pathogen, any kind of opportunistic pathogens also possible are there. Isn't it? So only the available drug of choice is immunosuppressive drug that reduce the overall immune attack. But recently, that means in the last January, I think so, yeah, the UK-based study released
Biscuit panma.
the information that we can use the corticosteroid, that is immunosuppressive corticosteroid, dexamethasone, control level. We can use the controlled use of corticosteroid dexamethasone. It has appeared to have a good positive effect on the severe cases of COVID-19. So the UK, they are using the control level corticosteroid, which will help to recover the patients from the severe cases. Yes. Here, I want to let you know one thing, is the NRF factor BIP is very important. So BIP is there, the binding immunoglobulin protein. This protein will be secreting during the stress time. So when the person infected by the SARS-CoV-2 infection, usually stress will be there. The infected cell stress will be there. During the stress, BIP will be increasing. NRF factor 2, isn't it? So this NRF factor play a major role, isn't it, to maintain the oxidative one thing. So what is the NRF factor? Nuclear factor erythroid 2 related factor. It's a basic leucine zipper protein, isn't it? This protein NRF factor regulate the expression of antioxidant protein. So the NRF factor play a major role to maintain the antioxidant, isn't it? So and also protect the cell from oxidative damage, so triggered by cell injury, cell inflammation by the viral particle. So if the person is without any complication, that is no comorbidity, so the infected people, he has the stress, but BIP will be higher and NRF is there, but that level is very low. So the normal cytokine production will be there and no severe cases. So in cytokine storm, if the person get the cytokine storm, so this kind of BIP, NRF2 factor play a major role that will also be associated to aggravate the cytokine storm. This is also possibility are there. But in the normal healthy people infected by the SARS-CoV-2 without comorbidity, so and the BIP protein and as well as NRF2 factor so play a role, but they won't affect our body. Normal cytokine production will be there, and then they can escape from the severity of the COVID-19 complication. But those who are in the comorbidity cases, like a cardiovascular problem, diabetes, and aging, so what will happen? The BIP level will be drastically increased. That drastically increased BIP, it will go for cardiac dysfunction, and the cardiac myopathy will also be possibilities are there. Then NRF factor will also be downregulated. This NRF factor is quite secreting more in the youngsters. If they have any cell injury, automatically the cell will be rectified by the secretion of oxidative proteins, and then maintain the oxidative reaction, antioxidant reaction. So oxidative damage will be rectified by the NRF2 factor. So now the doctors, they are using anti-stress treatment for PBA and PB125. This PB125 is nothing but the plant product, isn't it? So the for PBA is the phenyl butyric acid. These two molecules act as an anti-stress treatment. So we thought that in our university, when I presenting my recent research work, so I thought that maybe the capsaicin, so maybe act as an anti-stress treatment to induce act as a NRF2 factor to maintain the antioxidant protein and protect the oxidative damage. Isn't it? So if you have any idea, you can work on that. So how the carbaceramide, the product, the decoction, work on our lung tissue, whether it acts as a NRF2 factor to maintain the antioxidant proteins, isn't it? So this is the major one here. Now the once the people are hospitalized, so what kind of treatment they are giving? Many treatments they are giving. In the last year, they have given chlorophenone, hydroxychloroquine, to prevent the cell endosome formation. Isn't it? Though the viral particle attached on the cell surface, they won't form endosome, prevent the endosome. Now we are giving that. You know very well about the remdesivir. Remdesivir will block the RNA replication. Isn't it? So not only this, many vir compound is there, lopinavir, ritonavir compounds are there, but these are the compounds of supportive therapy. But other complications are also there. That is, immunological complications are there. To avoid such kind of immunological complications, right now we have the only one choice: immunosuppressive drug, anti-inflammatory. If you use anti-inflammatory steroid, that will also trigger the sugar level. So now, how to treat the cytokines stroke? Yes, that is the one corticosteroid. So the same thing I have mentioned here. Now in China, they have a clinical trial that is called tocilizumab. One drug is called tocilizumab. It's a monoclonal antibody. That monoclonal antibody will prevent the interleukin molecular activity. So when you use the monoclonal antibody, that antibody will prevent the interleukin 6. So no interleukin 6, no cytokine activities. No cytokine activities. The cytokine will be silent, will be muted. There is no further cytokine inducement. So now the China has already been approved the interleukin 6 inhibitor tocilizumab to treat the patient with the COVID-19, pneumonia, and as well as those who are having high level interleukin 6 level. So now many number of clinical trials are undergoing under the way to treat the cytokine storm cases. Isn't it? So now the positive results are that tocilizumab is the option. So not only this kind of the MAB treatment, now MAB treatment is using for all kind of cancer research treatment is going on, isn't it? Then our work, isn't it? So one recent research in January, Roda et al. From UK conducted longitudinal analysis of persistence of immune memory cells following the mild COVID-19 cases. After the mild case are those who are convalescent. That convalescent people elicits the memory lymphocytes that persist and display the functional hallmark of antiviral immunity. Now the research is going on. So based on this idea, based on this paper, we got an idea. Why don't we go for after taking the Sinopharm China vaccine, we can check the memory cell persistence of memory cells and the persistence of plasma cell in the vaccinated people. So what are the points that should highlighted in this paper? What they have highlighted? Antibodies are capable to neutralize the virus, and that antibodies will persist for at least 3 months in most subjects. So, and as well as the viral specific memory T lymphocytes are there, and T lymphocytes also display a hallmark of antiviral immunity. And then those who are infected, they have monoclonal B cells, and the numbers are increased, and that monoclonal B cells, they have expressed antibodies. That those antibodies are neutralized, and those antibodies are capable to neutralize the SARS-CoV-2 infection. So this is the one. Yeah. Now the conclusion. That's all. This is my presentation. This is the conclusion. So what is the take-home message of this presentation is there? So when a person is infected with the COVID-19 virus, the viral particle try to multiply in our body, especially in the upper respiratory tract region, and even in the lower respiratory tract region. Meanwhile, the B lymphocytes, humoral immune system, produce the antibody to block the infection. then the antibody bind themselves to the virus to prevent it from the infecting healthy cells, and also signal the other white blood cells to destroy it. This is the normal immune mechanism. If the antibody produced remain in the recovered patient's blood as immunity from future infection. So as a normal immune response, and as well as white blood cells, if they are aware of the viral infection, sure that immune responses will remain maintain their immunity for up to the immunity for long period to avoid the future infection. This is my take-home message. But cytokine storm also possibilities are that we should be study the cases and then try to discover, try to find out the solution to completely avoid the cytokine storm. The immunosuppressive drug is not a choice to suppress the entire things. We have to go for alternative one. That is the modern technology is called monoclonal antibody therapy. So for each individual interleukin, we can use the monoclonal antibodies are needed. So we have to establish our knowledge, establish our research to go for the new technology to find out the more monoclonal antibodies. So that's it. So this is our project. We got the approval recently. We got approval. We have sent this project to the Ministry of Health. Now under the process, they are asking many queries, not like other things. How we are going to collect the patient's blood sample, how we are maintaining. If anything goes wrong with the people, who will bear the cost, who will treat. These are the things are there. But Alhamdulillah, we got nearly. We have reached 90%. Once we get the approval, we can start the collecting the patient and the volunteers blood is vaccinated people blood. So our project title is Immunogenicity Efficacy of Sinopharm Vaccine Administered to UAE resident. This project will continue up to 1.5 years. So thank you very much. Thank you one and all to listen my lecture. If you have any questions, please raise your questions.
Thank you so much, sir, for this outstanding and thought-provoking presentation. You have explained everything in a way about SARS-CoV-2 infection. Now the topic is open for discussion. We greatly appreciate all the participants to come up with more questions, and our chief guest is pleased to answer them all. Any questions? So to start with, sir, shall I ask a question, sir?
Please.
Good evening, sir.
Good evening, sir.
Sir, my question is, this is Kishore from Hyderabad, Narayana Educational Institutions.
Yes, sir. Yes,
my
sir.
question is, can we expect more mutant varieties of COVID virus in the near future?
Good question, sir. $1 million question. Very proud of you, sir. Very good, sir. How we know, sir? How we know, sir? So, see, sir, within 1 year, lot of variety mutants are there. We don't know. That's why, sir. It's like a confusing, baffling. So suddenly there are some strains will be more severe, some are very less. We don't know, sir. That's why it's a complete catastrophic. And we don't know. We didn't expect this much. Possibilities are there, sir, but we don't know, sir.
Yes, sir. Because the more upcoming waves will be having more mutant varieties and everything.
No, sir. Actually, sir, the scientists also confused. Especially, I was attending one WHO meeting in last August. That is mainly funded by the WHO. WHO Chief Infection Control lady, she's from Netherlands. She said, we don't have any information. So we have only the hand sanitizer information, how to clean, how to sterilize the hospital information. Last year, there is no publication, sir. There was no publication. So entire WHO also very confused one. What will be going to be next? That is a big question. So nearly more than 4 or 5 months around last year, May, June, July, August, last year. So they were confused. They didn't get any data. But another one thing is that China, they have published a lot of data in the BMC journals. You can find out a lot of nearly more than 100, 200 publications are there. So all are they have stated and then found that genetic variations, minor variations, sir. It's the variations occurring in the clade level, not in the big region, the open frame region. 1/3 of the region there is no mutation. Only particular clade is there. That clade slightly changes their attachment capacity, especially RBD capacity.
Okay.
Yes, sir.
So I have a question, sir.
Thank you, sir. Thank you, sir.
Thank you, Mr.
Excuse me, sir.
Yes, madam.
Sir, actually, can we know about the after effect of this COVID infection? That is, after chikungunya, most of the record patients are suffering from knee. That is a joint pain, no, sir.
Yes,
So I like
after
that
the
because the patients are
chikungunya.
treated with, because in COVID also we are treating the patients with steroids. So is there any possibility of after effect, that is the occurrence of diabetes? That is because I have read an article that because of the steroids, most of the patients will be more prone to diabetes in future, like that. So
No, actually they are telling, but one of my known close relative, my sister-in-law, she affected by the COVID. She recovered. Alhamdulillah, she recovered. Very pathetic one. And one my close friend, he died just 2 days back because of the same cytokine storm. He got the microcoagulopathy, and then he got the cardiac myopathy also. He got the RS. He recovered. Afterwards, the microcoagulopathy started. So here, chikungunya viral may induce the antibodies. That antibodies or that any kind of immune complex may be related with viral particle. We don't know. We have to go for the study. But one thing, maybe we have to check with the chikungunya viral protein structure. That protein structure, especially in the form of epitope protein structure, and then SARS N protein or nuclear protein. If it is similarity is there, then this is called cross reaction mimicry, molecular mimicry. This molecular mimicry may also, the antibody may also react with this viral particle. Then after the antibody and then viral antigen, both are stored with one another. They can deposit in the joint region like immunological reaction. So what will happen? The deposition occurring in the joint region, synovial membrane fluid will be inflamed. So automatically the people will get the severe joint pain. Now one of my colleague, she is in the nursing side, she got the severe joint pain. This complication will be vary from person to person. So when I got the infection in the last year January, but mild infection. I got the bitter taste during the infection. One of my physiology professor, very close professor, is very close friend of mine. So he also infected by COVID-19. So he got the sweet taste. Even if he drink the water also, he feel sweet taste because some neurological changes are there. So how the receptor recognize that taste? So many research is going on. If you want, you can do the research also in that aspects.
Thank you, sir.
Yes, many things are going. Yes, ma'am.
Sir, I am Sita Vichalakshi. I am a class 12 student. Sir,
Class?
I have a doubt.
Excuse me, please. Which class?
Sir, class 12.
Yes. Great one. Yeah, class 12. Tell me.
Sir, why students below 18 years are not vaccinated?
Correct. Actually, the NRF factor will be higher in the youngsters.
Okay, sir.
And as well as, isn't it? And as well as the ACE2 receptor will be higher in the youngsters when compared with the older people. So we have to save the older people. So youngster people, even the viral particle enter, the ACE receptor will be more, and then maintain the angiotensin 2:1. So the angiotensin 2, it's a vasoconstrict. The blood vessel will be narrowing. So due to the narrowing, that means what I exactly tell me which means the ACE2 enzyme convert the AC to angiotensin 2 molecule to angiotensin 1. So maintain the blood pressure, lower the blood pressure. Isn't it? It's a higher, normally higher in the youngsters below 18 years, isn't it? But that data also be disproved because many 18 years people, 17 years people, they have BP and other things, especially in the Western countries because of the food habits, isn't it? Now a lot of people, they are diabetic. A lot of people are more stressed. A lot of people are more hypertensive in developed countries. Isn't it? So now the WHO recommended. So why don't we go for below? We can go for vaccination 12 or above 12. Now they have changed. The first priority of our government is the first priority of our government. We have to save the elder people, then above 40, then now they are giving vaccination above 18. But here in the developed countries, GCC countries, and then Western countries, now they are starting. They have started above 12.
Okay, sir. Thank you.
Thank you.
Good evening, sir. I have a question for you.
Yes, ma'am.
Sir, will we test positive for antibodies after COVID vaccination?
Yeah, antibody will be there because the vaccine is the viral protein. That is called dead viral particle. If you are taking Covishield or Covaxin, if you take Covaxin, it's a dead viral particle. Covishield is a recombinant vaccine, that is a chimpanzee adenovirus genome is a recombinant with our SARS-CoV-2 genome. So that vaccines are there, but lot of complications are there. We heard the literature, we heard the news, especially our vice chancellor from France is the immunologist, is a cancer immunologist. He stated a lot of people, they got the same complication after the taking vaccine, Covishield vaccine. But in our India, we are mostly Covishield is there, nothing happened. And then as well as the Covaxins are there. After taking vaccine, sure, the antibodies will be there. If no antibodies, what is the meaning of vaccine?
So can you,
yes,
sir, can you share some of the different effects, that is the positive effects of the different vaccines, that is we are using different vaccines, Covaxin, Covishield, Sputnik?
No, actually, vaccine is not a problem. The problem is our body is the problem. This is the main thing. Based on the obesity and then comorbidity, severe diabetes, or any other complication, any other allergic reaction, this is also possible because we don't have much more phase studies. It's a short period, they have developed the vaccine. Isn't it? So even there is no 10 years time. For example, like polio vaccine, like hepatitis recombinant vaccine, a lot of studies are there. They have a lot of period, they have spent time. But this catastrophic situation, we have spent only 1 year to develop different type of vaccines. So, and as well as many companies, they are giving the data, they are projecting data. Our vaccine efficacy is 90%, 95% based on the limited population. And also where they did in the same national people, sometimes it's not true. Isn't it? They go to the poor country and then select the people, voluntarily they are using, and based on the reaction, they are giving the report like that. Even we don't have much more data about many country vaccines.
Thank you so much, sir.
Welcome.
So
Sir,
any more
sir.
questions?
Hello, sir.
Yes, sir.
I have a few questions for you.
Yes, sir.
I congratulate the organizers and the speaker for their tremendous effort in spreading awareness about COVID.
Yes, sir.
In my knowledge, I come to know that no country is there with 100% vaccination. India is also in the queue. What is the minimal percentage of vaccination percentage needed to develop a herd immunity?
Yeah, at least, sir, it's based on the population wise. Sir. This is mainly statistical population wise. Like our India, at least more than we can go for more than 60%. At least reach the 50%, we can expect the herd immunity. It's mainly it depends on the population one. And how many elders are there? How many youngsters are there? In Western countries, elder peoples are more. In India, youngers are more. It will vary from country to country, sir. Herd immunity.
Okay, sir. Suppose if this question is giving an embarrassing condition for you, you can say pass, right? there is a belief in most of the people that this is a bioweapon. What is going on here is a bio-war.
Yes, sir. Exactly correct. Exactly
Agree with that?
Correct. Exactly what you said is correct, sir. See, each countries, they have the biosafety level 4, sir. When the COVID-19 started in the 2019 December, we heard the literature. We got the literature from the National Microbiology Laboratory, Canada. They have a lot of controversy over there. So what they said, so two Chinese doctor, I don't want to discuss here, but there is no option.
That is the reason why I said that.
This is a true or not. They say they developed the Wuhan Virology Institute, isn't it? They have given the Ebola virus, they have given the Rocky Mountain Spotted Fever virus, and then they have given the SARS virus, isn't it? So these two people they have donated there, and then many people they went there, they have developed the laboratory. So afterwards, the two Chinese doctors they were arrested. That Chinese doctor researcher, her name is, I forgot the name. She is a lady. She got the fund, very higher fund from the Canada for Ebola viral vaccine development. So many countries they are telling it's purely based on the economy. They want to be holding the leadership like that. We don't know, sir, what's going on in the inside. We don't know. That's why even our place also, because here we have the rights to discuss in openly in our university with the chancellor and other people. Even our research vice chancellor is from France. We have discussed these things, sir. So why the WHO is very silent? Who is funding the WHO? These cases are there because in the first wave, so much death was there in the US and then Italy, France also. So Italy, France took too much out there. And now the second wave is drastically moved to the India. So we don't know what is the third wave, like that. So, but the people are expecting it will continue up to 2025. They are thinking about that. It will continue, sir. It's a new normal. We have to adapt. We have to adapt with that. We have to adjust our life with that.
So you say that our lifestyle will not get changed until 2025.
Many people are expecting, sir. Even I'm not telling, sir, in the WHO discussion, because weekly once we are attending that webinar with a WHO sponsor, Nature sponsor, Thermo Fisher company sponsor. They are daily sending the webinar. Today you have to attend this one. How to diagnose this COVID-19 by using flow cytometry, by using NMR method. So many webinar we could not able to attend, sir. So important things we are attending. They are discussing this one. Many things are going on.
So another important question is: suppose once if one person is vaccinated, means how long the immunity will be there in his body?
That's our research, sir. I showed you, sir. This is our research. Actually, here two vaccines are available. One is a Pfizer vaccine. Another one is China vaccine, Sinopharm vaccine. The UAE government.
You can forget China vaccine.
No, sir. What I'm telling, everybody is blaming that one. Here, what I am supporting that vaccine, Covaxin as well as China vaccine, because those vaccines are dead viral vaccine. The dead viral vaccine, they won't create any complication in our body. But we don't know about the mRNA vaccine. And we can trust that recombinant vaccine also, that is Covishield. But companies, they are not providing the proper the phase trial studies. How many people they got vaccinated, how many people they got the complication, so how you treated that one, we don't have much more data, sir. Because this is the company secret, they want to sell the product. It's mainly based on the marketing, business and other things. How long they may be persist, even now we got the paper, recently we got the paper. There is no much more studies about the vaccination. After the recovery, the patient recover from the COVID-19, they may have immune power, especially memory cells, up to 8 months. So, like that, sir.
Thank you. Thank you,
sir. Thank you, sir. Thank you, sir.
So I have a question on behalf of our participants, sir.
Yes, ma'am.
Dr. Dania, is any particular mutant virus found to show excessive cytosine storm induction in infected patients?
No, ma'am. Actually, not only this, even if the people get H1N1 infection, along with that H1N1 infection, pneumonia infected, bacterial pneumonia, mostly or viral pneumonia, that is called viral pneumonia is there. So cytokine storm will be higher. Even in the autoimmune diseased patient, any autoimmune diseased patient infected by COVID-19, sure the cytokine storm will be higher. Even the people diabetic or any other complicated people, obesity people, aging people, sure it will vary from people to people. Person to person it will be vary. We don't have much more medical facility to monitor the cytokine storm. So now the developed countries, how they are measuring, particularly we have to measure the interleukin 6, interleukin 10, and then T helper cell 17 inducer key factor we can identify by using flow cytometry method or ECLIA, electrochemiluminescence immunoassay method. Now they are doing fully automatic measure. They are checking, but it is not available to the common people. It's not open market to all the people. This is the problem.
So people who got vaccines 2 times, do they have to wear a mask?
Sir, correct question, sir. Actually, the world is completely going in the wrong side. So when you use the mask, even the mask may contain the viral particle. Your nose may contain the viral particle, but it won't replicate in our body. Even it enter in our body, maybe 1 or 2 viral particle will replicate. That will be neutralized. But why the people are worried about the after taking vaccine, you should wear the mask, you should wear the mask? We don't know.
Sir, is there any study which explains the minimal load of viruses required to create an infection?
And there is no much more study, sir, because here the minimal load will be depends on the body nature. Your body nature. Body nature. So this is the one, sir. But we have a lot of studies for bacterial minimal load. For example, Vibrio cholera or Streptococcus or any kind of pneumonia. We have the minimal load we have. But this virus, we don't have a data, sir. So maybe later it will come.
I'm not asking these questions just for me.
No, sir. Correct questions. You are asking very good questions, sir. You are from which field, sir? Sorry, sir.
I am from microbiology, sir. I worked for Sadakathullah College. I'm proud to say that I was a member of faculty in Sadakathullah College.
Okay, sir. You are from microbiology, sir.
Yeah.
Thank you, sir. Thank you, sir. Nice to meet you, sir.
Good, sir.
Thank you, sir.
So another one doubt from a participant, sir.
Yes, madam.
So whether COVID will be spread from the COVID dead person? She's asking.
So the viral particle will not persist more than in the dead people. How long it will be persist is based on the viral load. Viral load is there. Only the nasal area. If any leakage, fluid leakage from the nostril, we can use any sterilizing agent, Dettol or Savlan. We can cover with cotton. That's enough. It won't spread unless the nostril fluid is there. If the viral particles are there, if the people touch the body, touch the nose, possibilities are there. But by touching the viral particle won't enter in our body. Immediately we can go for hand sanitizer and other thing. Washing is okay. Unless it is exposed in our nasal area or oral area, nasopharyngeal area, even in the conjunctiva, possibilities are there.
Good evening, sir. I have one more question.
Yes, ma'am.
What's about that white fungus, black fungus?
Madam, you are asking, Kavita ma'am?
Yes, sir. So
okay.
So can you say something about the white fungus, black fungus?
Ma'am, this is a name only, ma'am. Like Michael Jackson, like what is that? Arnold Schwarzenegger, like a James Bond. That's all are heroes only. All are heroes only. So usually this kind of fungal infection is quite common in those who are immunocompromised. Immunocompromised means not only the COVID patient, even HIV patient or severe tuberculosis patient or severe COPD patient, chronic pulmonary obstructive disease patient, COPD patient, and as well as post-operative patients or those who are taking immunosuppressive drug. Possibilities are there. Now they are projecting black fungus, white fungus, yellow fungus. Now they are sending the WhatsApp University black fungus enter. Then we will get the gangrene like a black color. It's a complete humbug. Possibilities are there, but mainly the fungal infection main reason is climatic condition.
Yeah. Because of the
Main one is climatic condition. So we could not expect the same fungal infection in the Western countries. Those who are in the polar region, we cannot expect. Especially in the Delhi and then metropolitan city, and the pollutions are there, more depressions are there. Due to that depression, the fungal growth will be more.
Okay. You can ask,
Hello, good
ma'am.
evening, sir.
Sir?
Hello, good evening, sir.
Yes, sir.
Yes, sir.
I am Harinathan from VHSN College, Nagar, sir.
Yes, sir.
You already explained about the drug called Remdesivir. What
Yes,
about the mode of action of the drug called Lipnovir, sir?
Ritonavir.
Yeah.
Ritonavir is for other things, sir. They are not using
Lipnovir, you told, sir.
Lipnovir.
Yeah, yes, sir. In your presentation, you have mentioned, sir.
Yes, sir. Here. Yeah. So, can you? I will show my presentation. Show my screen.
Yeah, okay.
Yes, sir. Can you see this one? Yeah.
Yes, sir. Yes.
Yeah. So the Lopinavir, so
yes,
involved to arrest the production of viral protein, subunit viral protein. So there are two, during the viral replication, there are two pathways are there. One is yearly pathway and the one is secondary pathway, late pathway. The primary protein, secondary protein. The primary protein will arrest the viral, the infected cell. Then secondary replication will start to synthesize progeny viral proteins. That protein will be blocked by the Lopinavir, and also if the protein already synthesized, to block the attachment of those protein in the endoplasmic reticulum. Endoplasmic reticulum play a major role to carry the protein and form a vesicle to produce or to form a progeny virus. So this is the one, sir.
Okay, thank you, sir. Thank you, sir.
So
thank you for your presentation, sir.
Yeah, thank you very much, sir. Thank you, sir.
Thank you, sir. Thank you.
So another question from the same participant.
Tell me, ma'am.
One of her relatives had COVID, and after 15 days, he returned home from the hospital. Now, whether he has to be in home quarantine or else?
No. After the recover, once recover, they have to check the PCR. If there is no viral particle, no need to go for quarantine.
Okay,
the PCR is very, very excellent. But before going for PCR, they don't go for steam vaporization. The steam vaporization try to remove most of the viral particle, even though they are the positive. Then after taking the steam vaporization, then what will happen? They will get the pseudo negative, the false negative result will come.
Okay.
Many people are doing like that. In India, many people are doing like that. Maybe they may give the result, positive result, then immediately the government will take us and then make it in the quarantine, how they have done in the last year. Like that, many people are thinking and then worried about the government approaches, and then neighbors also. So what they are doing, what they are practicing, before going to test the PCR, most of the people, they are using the steam vaporization. And this is the wrong approach, isn't it? So what will happen?
Actually, it is highly recommended here.
No, we tried this one. We tried in our patient. We tried in the studies we are going here. One more studies we are going here. If the people are positive people, positive, home quarantine. So okay, we can go for the steam vaporization. After taking steam vaporization, within 1 hour, when we check in the nozzle, the PCR is negative.
Okay. So it is recommended as a precautionary measures.
No, they can go for PCR test. If the PCR test is negative, it's okay. They're all right.
Okay, so another question, sir. So, is there any chance of getting bacterial pneumonia as cross contaminant with this COVID?
Pure possibilities are there. That is the many people are there. Many people are died due to the bacterial pneumonia. First they got the bacterial pneumonia. Even viral pneumonia possibilities are there. Because seasonal is there. You know, usually in our Tamil Nadu during the rainy season, adenoviruses are possible. That is called medrasai. So not only the adenoviruses also causing the respiratory infection also. And gastric infection also. Many viruses are there. If the viral enter along with the corona, other viral particle enter, that is a co-infection, then easy to get our own flora, oral flora, staphylococcus possibilities are there. But luckily we are in the home quarantine, we are wearing the mask, so we can avoid most of the bacteria.
Good evening, sir. I have one
Another
question for you.
Okay.
Proceed, ma'am.
Sir, is herd immunity possible with COVID?
Possibilities are there, ma'am, because how many people are infected, how many people are recovered, how many people are died. This is the main one, ma'am. If the people are mildly infected, asymptomatic people recovered, that people, we don't know how long the immune cells immunity will be persist in his body. Maybe 1 year. Now the recent studies they are telling up to 8 months it's there. Then another studies will come. How long this herd immunity will be there? We don't know, ma'am.
Sir, can we think that COVID is zero at China now?
Sir, we don't know the report, sir. We don't know the report. That's why. But many people are saying how the China they have control. They have control, sir. Strict quarantine. Strict quarantine. The people should be strict, very quarantine. We can control, sir. Now that's why, sir. New Zealand there is no cases. Australia, one case started because of my friend son is studying there. One of my close friend is working in the pathology department. We were very close. He said my son is there in the Melbourne. One cases they have found, immediately that person really arrested, and then put it in the separate quarantine. Then 1 week they have completely locked down. They put the lockdown. So like that we can maintain, sir.
So for countries like India, it is not at all possible. We cannot compare our status with New Zealand or Australia.
Because of highly populated ones, sir. Another one is what about the daily wages? Where they will go? How the government will provide the money and other many things are there, sir. This is the problem. So we expected last year, if it is going wrong, it is very difficult to face the catastrophic situation in India. But last year we escaped, sir. This year is more, sir.
So you may be having some idea about the infrastructure facilities connected to the public health in China and India.
Sir, this is country-wise, sir. Country-wise, and as well as the China will sell the product. Economy.
My question is not over. How many marks will you give for the Indian health infrastructure system compared to China out of 10?
China is more infrastructure is more, sir, because they have occupied entire Africa. They have occupied the entire Middle East area for selling their product. So they have a lot of money. Based on the money, they can develop anything, sir. They have a lot of money, sir. Now they are funding WHO, sir.
What is your mark?
We cannot tell you, sir. Otherwise, because we don't know what is the infrastructure is going on in India. I'm here. But I know about that both things. But Chinese, they are maintaining because of rules, sir. Very strict rules they are following. But India, no, sir. If the government implement 2 more lockdown, the people will get angry, and then they immediately post and criticize the government. People should change, sir. People should change. Don't blame the government. People
very difficult to come out of that.
Many people are, sir. Many people. Yesterday, my close friend, he died. Day before yesterday. He is a very good. He is a normal. He doesn't have anything. So during the infection, his sugar level raised up around 400 something, and then pulse rate also fluctuated, but maintained. Suddenly, after 2 days, he thought that he will be all right, and then he will be discharged within 1 or 2 days. But suddenly he got the microcoagulopathy because of Cytokinestone. We could not expect, sir. We could not because these are the things are silent ones, sir.
Thank you so much, sir. Thank you so much for the excellent answer.
Thank you, sir. Thank you, sir.
Okay, sir.
Okay, ma'am. Thank you, ma'am.
One last question. Sir.
No problem.
Many have asked a lot of questions.
No problem.
It's possible for mosquitoes to be a vector for transmission?
Pardon?
Is it possible for mosquitoes to be a vector for transmission? That is transmission of the virus.
Madam, already we are in deep trouble.
No,
Don't invite
mosquito.
mosquito.
No, not possible, ma'am. Because mosquito, the viral mosquito Aedes aegypti, if they come mix with the Aedes aegypti viral agents or yellow fever viral agents, possibilities are that we don't know. We don't know, sir. We don't know. Don't give this idea to people. No, because sometimes the people will try, maybe like that.
Okay, sir. Okay, sir. Thank you so much, sir.
Thank you,
You have
thank
cleared
you.
all the doubts of all the participants, and we have pleasure to have you today with an extensive knowledge about this immunity against SARS-CoV-2. So now the next session is the vote of
No,
thanks.
I need some feedback from your things.
Sure,
Make
sure.
a feedback and then send it to me, ma'am, through
Sure, sir. Sure,
how
sir.
they expected, how was the presentation.
Sure, sir. Yes, sir. So may I request Dr. Manohar, Assistant Professor of Microbiology, Sadakathullah Appa College, to thank the gathering.
Am I audible?
Yes, sir.
Yes, sir.
Good afternoon to all. This is my bound duty to express my sincere thanks to Almighty for making this occasion grand manner. On behalf of our management, I thank Principal Mohammed Sadiq sir, Vice Principal Syed Mohammed Raja sir for given permission to conduct this one-day international webinar on transmission of pathogenesis of SARS-CoV-2 and its immune response. On behalf of our management, principal, our department, I express my sincere thanks to Dr. Nazirullah Rahmatullah for giving a wonderful lecture. I think the students are with more energy. If you like enrich our knowledge regarding the COVID-2. Thank you, sir.
I would like to.
I express my sincere thanks to.
My sir is there. My teacher is there. He has joined. Dr. Syed Jahangir. So
welcome, sir.
Welcome you, sir.
Welcome, sir.
Dr. Syed Jahangir from Jamal Muhammad College. He was my teacher. Very close teacher. He is my like a brother. My advisor also. Sir, can you hear me?
Nice.
Yeah, yes, doctor.
Yes, sir. Can you open the video, sir? I want to see you.
I open it, but not get it, man.
Okay.
It's all right. It's all right.
Thank you, sir. Thank you, Mr.
But you did well. You did well. Congrats.
Thank you, sir. Thank you, sir. Sir. Thank you, Mr. Because of
Okay. Please carry on.
Because of you, I am here, sir.
No. God is great.
God is great. That's you help me.
No. God is great.
Yes, sir. Thank you.
This is for your hard work and dedication in the field of microbiology.
Thank you.
And you bring a lot of to our department as well as college. And I especially thanks the management, principal, and the organizers of Sadakathullah Appa College for your wonderful the need of the hour regarding the COVID. But you did it very well and give a very excellent knowledge regarding this one. Thank you very much. Congrats.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you, sir.
I want to tell you
one
thing.
Sir?
Yes, sir. Students are there, sir.
Okay. You proceed, sir. You proceed, sir.
Actually, I want to tell you the one thing. I have a good relation with the Sadakathullah Appa College, not in the college, Syed BD company.
Okay.
Because when they doing my MSc. Hello, sir. Am I audible?
Yes, sir.
Yes, sir. So when I was doing my MSc PG, that time I could not able to fees also. Yes, that time fees was very less. Yes, sir. That time my family was very poor in condition. Financially, we are very crisis. That time I approached the principal, Jamal Muhammad College principal. He said, we will arrange something. They have arranged a part-time job in the Syed BD company. They have given monthly nearly 750 rupees. I have a good relationship with that people. During the exam time also, I didn't attend many. I nearly 15 days I didn't go to the job, but they have given the full salary. That time I said, sir, I didn't attend the full month. I have attended less than 15 months. No, we are giving for your scholarship, like a scholarship, but we don't want to give us a free. We are expecting something. Then only you will realize this is your money. So I want to tell you this time, but I didn't expect I am here. I am today. I am giving the lecture to the Sadakathullah Appa College. So I thank the management. They are helping a lot of people like me also. So my dear students, please concentrate on your studies. And now the situations are very worst, but don't divert your knowledge. You have to update your knowledge. So if you gain knowledge, the world is yours. You can reach even in the moon or even in the Mars. Now the next project is going on in NASA. My dear students, you should know one of the NASA project is there. Her name is Shalini or Shantini Iyer. She is from Tamil Nadu. She is the chief coordinator for the next moon, the human moon project, and as well as a human Mars project. She is the lead faculty. So, my dear students, you have to read. Don't think it out. This is waste. This is waste. You have to read, and then gain more knowledge. One day you will be achievable. Thank you, sir.
Shall I proceed, sir?
Thank you, sir.
So I express my sincere thanks to our faculty members of department, Dr. Janet Ma'am, Dr. Chitra, Dr. Feroz Khan, Dr. Senthil, Mr. Sharif, Dr. Surya Prabha, and Dr. Kavida for your valuable support for this one-day international webinar. Thank you. And last but not least, I express my sincere thanks to the students and faculty members from other colleges and our colleges for participating in this one-day webinar. Thank you, Ananda.
Thank you so much, sir.
Thank you.
So we extend heartfelt thanks and wishes to you all who participated in this webinar by patiently listening and questioning. So expecting your participations in future webinars also. Let me conclude here. Thank you so much. Thank you once again. Have a great day.
Thank you, ma'am. Thank you very much.
Thank you so much, sir.
Thank you, sir.
Thank you, ma'am. Thank you.
Sir, very clear presentation. So நீங்க வந்து எங்க இருந்தீங்க அப்ப அதுல வர்க் பண்ணீங்க? You are in Tirunelveli or Trichy?